So embryo development and blastocyst formation. So we have stopped till fertilization. After fertilization, the fertilized zygote will get converted into embryo, and there is a structure called as blastocyst. This blastocyst will go and get implanted in the uterus. So let us see how the blastocyst is formed. What happens to the zygote till blastocyst formation? How the zygote, that is single cell, is converted into blastocyst. So first, embryogenesis is the development. Embryogenesis. Gametogenesis is the formation of gametes. Sporogenesis, formation of spores. So similarly, embryogenesis is the development of embryo from fertilized egg. How the fertilized egg is getting converted to embryo, and then from embryo it has to get converted into full-fledged organism. Full-fledged organism from embryo to fetus. That is called embryogenesis. Next point, the major stages. The major stages are, first is cleavage. What is cleavage? The division of zygote. Zygote divides into 2, 2 to 4, 4 to 8. That is called as cleavage. In simple terms, it is division, cell division of zygote. First is cleavage. Then there is blastosis formation. Many cells of the zygote will form a structure called as blastosis, which will go and get implanted. Then implantation. After the formation of blastocyst, this will get implanted into the uterus endometrium. That is called implantation. So as the cell is dividing, as there is cleavage formation, from 1 it becomes 2, becomes 4, 4 becomes 5. The zygote or the embryo will come down. Where it is now at present during fertilization, it is in the fallopian tube. It is inside the fallopian tube. From fallopian tube, it has to trickle down. It has to move down in the uterus, and it will get implanted in the uterus. Then there is gastrulation. There is a structure called as gastrula, which is formed. Just like blastula, there is gastrula. Blastosis formation means there is a structure called blastula. Gastrulation, there is a structure called gastrula. And then organogenesis. What is organogenesis? Formation of different organs in the embryo. So this is the structure. There is first fertilized egg. From fertilized egg, you will get 2 cells stage. Okay, 2 cells. Then 4, 8, 16, and then this is the structure. And the full baby will get developed by 290 or 288 days. Then let us see what is cleavage. Cleavage or segmentation or division of the cell or the zygote. So repeated mitotic divisions transform zygote into hollow, spherical and multicellular blastula. Cleavage is nothing but repeated mitotic division. We have studied mesosomatic and permanent tissue. Mesosomatic tissues are the tissue which has the ability to divide very fast. Initially, it will divide very fast. Then it will get converted into permanent tissue. So these are something like mesosomatic tissues. They keep on division, repeated division, and it transforms the zygote into a hollow, spherical and multicellular blastula. That is what is blastosis. Interphase in cleavage is very short. Usually the interphase is very large. In mitosis or in cell division, the interphase is large, but here it is very short. Cleavage does not result in growth. Cleavage will not result in growth. Growth means increase in size and increase in speed. It only forms new cells which are very close to each other. They are very close. There will be new cells, but they are very close, like compact. That's why this is called as compaction. It will not grow in size. They will keep on dividing and the cells are very, very close to each other. So that is compaction. Now here the fast DNA synthesis. DNA is synthesized very fast and more nucleus to cytoplasm ratio. More nucleus to cytoplasm ratio means the size of nucleus will be larger as compared to the cytoplasm. Because it keeps on dividing. When one nucleus is formed, a small cytoplasm will be formed and then it will continue to divide. Here you can see first zygote, it gets cleavage and create cell state, and it gets cell state is called as modula. From positive, there is desiccation. And then next is cleavage of human zygote. How the human zygote gets divided. First cleavage occurs in 24 hours. After 24 hours of fertilization, there will be first division. Second cleavage will be after 40 hours. So by this time it is around how many cells? 2, 4. First division you will get 2 cells. Second division you will get 4 cells. Third cleavage or third division is after 3 days of fertilization. After 3 days. So here it will be around 8 cells. 2, 4, 8. Then subsequent cleavage in orderly manner. Means it will keep on dividing and it will form around 32 cells. From 8 it will become 16. Then 16 it will become 32. And then what you get is called morula. The embryo is around 30 morula. Okay. Then embryo slowly moves down. After morula stage, from the fallopian tube, it will slowly move down. It has to come to uterus. Okay. So here you can see 2 cells, then 4 cells, 8 cells, 16 cells, 32 cells, morula. This zona penicilla is still there. Inner cell mass and trophoectoderm. Here you will see morula is around 16 to 32 blastomeres. 16 to 32 blastomeres. Now I have question. Blastomere. What do you think blastomere will be? The cells. The cells of the zygote which are formed after cleavage are called as blastomere. Cells produced during cleavage are called as blastomere. So morula is made up of 2 things. Inner cell mass and the trophoblast. Inner cell mass and the trophoblast. Inner cell mass is nothing but group of large centrally located cells. Some cells they will go in the center and get arranged there as a group, which are very large. That is called as inner cell mass. So you can see the structure. Trophoblast will not take part in the formation of embryo. Trophoblast will not form embryo, will not take part in the formation of embryo. The inner cell mass will take part in the formation of embryo. Next is blastosis formation. So from morula, from zygote, we get morula, that is around 32 cell stage. From morula, we will get blastosis. Okay. Morula enters the uterine cavity. Morula will enter the uterus. There will be a cavity. And uterus secretes a glycogen-rich fluid. Uterine milk. It is called as uterine milk. So morula is entering the uterine cavity. There will be a cavity. There will be a pit somewhere in which the morula will enter. And uterus will secrete a glycogen-rich fluid called as uterine milk. There will be a liquid which is secreted. Now this liquid that is uterine milk, enters the morula. Enters the morula. This uterine milk will enter the morula and gets collected between the cell mass and the trophoblast. Between cell mass and trophoblast, it will get collected. So what will happen in between the cell mass and trophoblast? There will be a cavity. Yeah, there will be a cavity. Because there is cavity, that's why it is called as blastocyst. Okay. Leads to a formation of blastocyst cavity, which is called as blastocyst. Seal means cavity. There is one group called as Coelenterata. Coelenterata, because they have a body cavity. That's why that seal word is come, blastocyst. So this leads to the formation of blastocyst. Okay. With the appearance of blastocyst, lorula changes into blastocyst. Once there is appearance of this cavity, after there is cavity inside the morula, we don't call it morula, we will call it as blastocyst or blastocyst. Inner cell mass pushes on one side. Inner cell mass, which was at the center, will get pushed on one side and form a knob called as embryonal knob. This is the embryonal knob. And in this picture, you'll come to know. See what is here. You can see how it is happening. It will form connection with the mother. What is that connection called? Placenta. It will form connection with the mother. The trophoblast will not take part in development of the embryo. It will help the embryo to develop by taking nutrients from the mother. It will form a connection. See, here you can see. This is the placenta. There are two layers formed by one layer is called as chorion, the other one is embryo. Chorion will form connection with the blood vessels of the mother. Chorion and embryo. Okay. Trophoectoderm or the trophoblast do not take part in formation of embryo. It divides into two layers called fetal membranes. This layer of chorion and amnion. Amnion is the amniotic fluid which surrounds the baby, which prevents the baby from shock or any pressure. Small hole is created in zona pellucida. Okay. Now that blastocyst which is covered with zona pellucida, no, there will be a small hole. It will come out of the zona pellucida. What is blastocyst made up of? The outer trophoblast or trophoectoderm and the embryonal knob. Now this trophoectoderm will form the connection. If there is zona pellucida, there is no connection. Zona pellucida cannot get much uterine. Only the trophoblast, which is the end, will get one. That's why it will come out from the zona pellucida. Okay. Then last part is implantation. Implantation is attachment of the blastocyst to the endometrium. Okay, endometrium, mesometrium, endometrium, that layer. Blastocyst sink in pit. There is a pit in the uterus where it gets sink and it's completely buried. Trophoblastic or chorionic villi. The trophoblast, which is the outermost layer, will form chorionic villi. Villi, you all remember? What is villi? Finger-like projection in the stomach. That is villi. Here they are different. Chorionic villi means like this, zigzag, like mountains. This will form connection with the endometrium so that nutrients can be supplied to the developing embryo. Then this is very important. Progesterone is the hormone which helps in implantation. Progesterone hormones helps in implantation. It secretes two substances called osteochondrin. There are two substances which are like glue, which will help to attach the blastocyst to the uterine wall. And end of implantation leads to pregnancy. Okay, end of implantation means it is pregnancy.